My kids are getting really funny. They're so used to muting that they'll.
Great. Hi, everybody. This is Dana Hargunani, Chief Medical Officer for the Oregon Health Authority, and we are here today for the OHA COVID-19 webinar series. I am joined by 3 colleagues today, a new addition to the team, which is fantastic. So I'm going to pass it off for everyone to introduce themselves. Go ahead, Tom.
Yeah, this is Tom Jeanne, Deputy State Health Officer and one of the Senior Health Advisors.
I'm Ariel Smits. I'm the Medical Director for the Health Evidence Review Commission, and I work off and on as a Senior Health Officer.
And my name is Joe Sullivan. I'm previously a family doc, now a Senior Health Advisor working on vaccines with OHA.
Great. We're really excited for today's presentation. I am going to see, there we go. So we're going to do our quick EPI update to start. Then we're going to go into a deeper dive around current situation with testing, give you an overview around vaccine update for COVID-19, followed by some just brief updates on the status of influenza and vaccination here in our state. Finally, closing with a literature update and answering some of your questions. As usual, you can send us your questions. I will try to broadcast them, and we'll try to answer as many as we can throughout today's webinar. So just to get us started with the EPI updates, here are the stats, if you will, since yesterday. We had over 38,000 total cases captured as of yesterday, with 2,800 hospitalized cases. And unfortunately, we have now surpassed the 600 mark of the number of deaths in Oregon. Couple things, I'm not going to go into too many more slides related to this recent EPI update, but a couple things I'll comment on. As usual, we continue to see the highest case rates among the ages of 20 and 29, followed thereby with ages 30 to 39 and 40 to 49 age groups. Additionally, as we share with you almost every time we speak, we continue to see disproportionate impact based on race and ethnicity. Still highest rates among those that identify as Latinx, Latino, Latina, Pacific Islander, American Indian, Alaska Native, Black, African American, and African immigrants. Hospitalization and death rates certainly do, though, increase with age. 50% of the deaths up until now have been among persons at least 80 years of age, and 75% among those who are 70 or older. And as you can see here on the EPI curve, we continue to see an upward trend in terms of transmission and cases here in Oregon. On this next slide, when we take a look at the last EPI week from October 5th to 11th, this is what we report in our weekly report published every Wednesday. So during that time period, we had 2,400, just over 2,400 new COVID-19 cases recorded. This is up 18% from the week prior. And the number of Oregonians newly tested was 28,490, up 26% from the week before. You might recall that we had fairly low testing numbers overall, particularly around the time period of the large wildfire impact in Oregon. Over the last week, we had 6.4% of test results positive, which was a slight increase from the week before. And just a reminder, these numbers may change and are constantly updated on our website. They are based on specimens collected during this time period. So as we get reports of those test results, these data will be updated accordingly. We just wanted to share this additional slide just to make sure you are aware that it's existing on our website. It's at the bottom. You can find it's on our coronavirus website under data dashboards. And this is the dashboard related to trends in test positivity, not one that we've shared with you frequently on the webinar, but is a useful one to look at. So you can see on the bottom left is the trends in test positivity statewide in Oregon. Again, we're sitting at about 6.5% right now. And then you can look at the trends across every county, or as you can see, the tri-county area in that second box. Also, if you're on this dashboard live, you can hover over each of these counties and get more information about testing that has occurred in that particular county and trends in the test positivity. As you know, test positivity has multiple factors that inform it, including the rate of testing that's happening, the rate of transmission of the infection, outbreaks that are occurring in that area, for example. So we invite you to take a look at this as we monitor this regionally as well. And I am noting a comment that the sound cut out. Just give a pause for a second. If someone doesn't mind dropping me a note that the sound is working, if you can hear me, please let me know. Again, if you are able to hear me and have access to dropping a note our way, we're trying to fix it on our end, but if you do hear us, please do let me know. Okay, thank you. We did see several people saying they can hear, which is great. We're sorry this occurred at our last webinar. It sounds like someone who had a problem was able to log out and log back in. So if you do find yourself to be in that position, maybe try that yourself. So sorry for the inconvenience. We'll keep going here. Finally, on the last part of this EPI update, we'll just share a relative information that we find important to track with you all is, and that's the COVID census across our hospitals statewide. This is reported daily by our hospitals in Oregon. And as we've been sharing, this has been also an upward trend over the last several weeks, backing out to early September. Our hospitals report that they remain very busy. The majority of their patients are not COVID patients. They've also been seeing shorter admission timeline for COVID patients than earlier in the pandemic, and less reliant on ICU level of care, as you can see in the bottom line, as well as ventilation need, a mechanical ventilation need. But certainly we're seeing an uprise trend in the number of COVID patients requiring hospitalization and in our hospitals right now. Moving on, we're going to pass it to Dr. Jeanne for a testing update.
Thank you. Yeah, so we have some significant updates on testing here to go through. The first is our testing guidance for healthcare providers, which was updated last week. So maybe some of you have seen this already. It's not I guess it's a significant change. It's mostly a change in terms of our wording for who to test. And so we've got an explicit recommendation now to test close contacts of confirmed or presumptive COVID-19 cases, regardless of whether they have symptoms. And then we also have a little information there you can see about the optimal time of testing, which isn't really known, but we are recommending a fairly broad window of 3 to 14 days after exposure, based on the data on testing sensitivity. Testing somebody right after exposure within a few days is not likely to yield a positive test. Just the sensitivity based on the natural course of infection is going to be very low. So we recommend waiting until at least day 3 after the first exposure before testing. And then we put this in just to remind everybody that a negative test does not change the need for a full 14-day quarantine for all contacts. We also have people exposed to COVID-19 in congregate settings, and migrant and seasonal farm workers upon arrival in Oregon, as recommended explicitly to be tested in all situations. We also have some information on interpretation of testing results that we've added to beef that up in this document and to help providers understand what a positive or a negative result from a various kind of test might mean. And then I guess the highlight from that would be that a negative test result, regardless of whether it's an antigen or a PCR or molecular test, doesn't really ever rule out COVID-19, because the sensitivity, even of the sensitive PCR tests, may be only as high as 80%. Okay. So an update on our BinaxNOW, and as a reminder, BinaxNOW is the test produced by Abbott. It's a rapid antigen test, and the federal government purchased, I believe, about 150 million of them, and is distributing them to all states. So we, you can see our allocations here. We actually got an early distribution of 15,000 tests, which was related to the wildfires for areas affected by wildfires. We've now received 2 full weeks of our distribution, 82,000 the first week and almost 62,700 on the second week. And we've distributed at least 48,000 tests so far. That's 23 counties in every part of the state, 5 Indian tribes or groups, some clinics, including Winding Waters Clinic, which is in Wallowa County, and the NARA Native American clinics. And then we have an additional 250 tests sent to long-term care facilities so far related to wildfires and shelters. So we've spoken with all counties in Oregon. We expect to ship tests to additional counties and tribes in the coming week. We're also reaching out to K-12 school-based health centers, student health centers at colleges and universities, and we plan to send tests directly to their clinics to help them with testing of symptomatic students and staff. And the distribution plan includes 33 FQHCs, 1/3 of which are in underserved urban areas, and 32 rural or critical access hospitals. Moving on now a little bit, we had some questions in previous webinars about what we're doing to help ensure access to testing for populations disproportionately impacted by COVID. So I've got a few slides on this. First, we are working with local community partners all the time. That includes faith communities, clinics, hospitals, schools, tribes, nonprofits, and other community groups to really ensure that our response is meeting the needs of various communities. We host bi-weekly community partner calls where it's really a two-way conversation with partners to share information and hear their questions in real time and kind of have closed-loop communication on queries and requests. And those are done in both English and Spanish. We also have daily community engagement coordination meetings where we discuss our coordination with various community-based organizations. And so here's some of the highlights from that. We're really trying to ensure that our testing materials, information, and services are culturally and linguistically concordant, ensuring that PPE that CBOs have is appropriate and standardized, and ensuring that wraparound services are appropriate and available to those who need to be tested. We're also working on state-to-state coordination for the quarantine of contacts of cases across state borders, and that would especially apply to families and contacts of migrant and seasonal farm workers from California. I've already mentioned our testing guidance, and that really does ensure testing and encourages testing for anyone with symptoms in consultation with their healthcare provider. And then we have a specific prioritization there for settings where resources may be limited. And that includes several of these groups that would be at higher risk, including people of color, people with disabilities, people whose first language is not English, and then people in congregate settings and frontline service workers. And then this is another really important thing that OHA does is we do support community testing events. We've had 10 events since August in several counties. These have been at work sites, community sites, faith-based sites, and one school so far. We've tested a total of almost 900 people in these events so far, and we actually have 20 more of these community testing events scheduled for October and November. And then finally, we have a COVID-19 test site locator that we published to help people find testing sites. It's an interactive map. Pages are available in both English and Spanish, and it can be toggled into other languages. And then a final note that the governor did reach an agreement a while back with insurers so that nobody should have to pay out of pocket for COVID-19 tests. And that would include the people covered by column, OHP, or private insurance.
Thank you, Tom. There is one question that came in that I wanted to pause for. So the question is, in view of the false negative rate and expectations or observations on Binax tests, what is the appropriate community messaging to be? I'm presuming
About a negative
regarding
result.
regarding a negative result.
Yeah, so that's a great question. First of all, as I kind of mentioned already, both PCR and antigen tests can have false negatives. So a single negative test does not rule out COVID. If you have a negative antigen test, because they are generally a little bit less sensitive than PCR or molecular tests, if you have a negative antigen test in somebody for whom there is high clinical suspicion of COVID, they have symptoms or they're exposed, for example, providers can certainly consider retesting using a molecular test like PCR, and also testing for additional respiratory viruses like influenza. So that decision really depends on whether, what else is circulating, so flu season, for example, available resources, and whether the test would alter medical decision making.
Great, thank you. A couple other questions, and I'm seeing still at least one person still having problems with sound. So we will try to do some quality improvement over this section and future sessions. We apologize. So one question is from someone who works in a community health clinic in Benton and Lane counties. They have 6 clinics across those 2 counties and are in FQHC. They haven't heard anything about receiving BinaxNOW tests and wondering when the county might be hearing about any tests coming their way and how that was determined.
Yeah, I don't know exactly how the process has worked in terms of which counties are contacted when. I believe if you have any connection, I would imagine talking to your local public health authority for the county that you're in would be your first step. And they are definitely connected with us already, and they can help you get linked and get testing.
And I would just add, I believe these conversations are just now all been rolling out in recent. So if it may be that your health department has more recent information. So just all of this has been fast and furious as we've been collecting many tests from the federal government. All this, the communication is rolling out as we speak.
Yeah, we've ramped up the process really fast. I think this week we're scheduled for our 3rd week of delivery from the federal government, but we're still, in the process of making sure that we're getting things to all parts of the state. So that process should continue to ramp up in the next week or 2.
A couple other questions, and then we'll move on for a little while for vaccine. You mentioned the agreement made with insurers about testing, not requiring payment or out-of-pocket costs. The question here is, do uninsured patients have access to free COVID testing?
I believe that is the case. I know, yeah, so my statement did focus on insured patients, including OHP. I'm not sure how the column status affects it, for somebody who might not have insurance, but for all comers who don't have insurance, I'm not sure that's a blanket free for everybody, unfortunately.
We can come back around and see if we can find more information to share, for sure. That is a great question.
But thankfully, I think we're still at around 95% of Oregonians who do have insurance. So it's just a matter of trying to close that gap.
Great, and maybe one more question, maybe 2 related. It says, do you anticipate getting enough tests in the future that anyone can get tested? I have patients wanting to visit elderly relatives over the holidays, but wanting to ensure they are negative before exposing themselves to these relatives. And then a related comment says, given the false negative rates, for example, of the BinaxNOW, we shouldn't fool ourselves or the public that this is a good testing strategy for asymptomatic people because it could be misleading. Right. So Tom, do you want to comment on that?
Yeah, I'll start with the second one. That's a very, very good point. That's something that we've tried to highlight. We're kind of swimming upstream against the mainstream narrative, including from parts of the federal government that would like this to be a screening test for asymptomatic people. The BinaxNOW, and in fact, none of the antigen tests that have received FDA EUA has been validated in children or asymptomatic populations. We would really like to see some studies to understand the performance of these tests in asymptomatic populations, but have not seen that yet. And then the first question was,
I think they were related. I think you've covered it.
Okay.
Maybe one last quick one, and then we'll turn it to vaccine, and we can come back if we have time, or certainly in our next webinar. So quick question, are the BinaxNOW results reportable to the state?
Yes, the problem is they're done point of care, and they're just on a card, and there is that app that's associated with them, but the app does not report to public health. So it's really, yes, they're reportable. They need to be reported just like any other test, any other point of care test, to the state, and that can be done through our website. There's an electronic reporting option. But that is one of our concerns, because since it doesn't go through a laboratory, it doesn't produce an electronic lab report, which is the way we get most of our test results.
Great, thank you. Okay, some really good questions coming in. We will come back to them if we can, either today or certainly at a future webinar. So I'm going to pass this now to Dr. Sullivan to walk us through some updates on COVID-19 vaccine.
Okay, thank you very much. It's a great opportunity to talk to you about a vaccine update. As Dana mentioned, things are fast and furious, and in the realm of vaccines, it's no different. You will be hearing about new vaccines coming to the market soon, probably, and you will also hear new vaccines going into phase 3 trials. But before I start, I want to give some context and a definition. So the context is, during this talk, I'm going to be talking about manufacturers' names. And normally in a medical talk, I would not do that. But this is how you see the vaccines in the newspapers. So the AstraZeneca vaccine, or the Pfizer vaccine, or Moderna vaccine. So what I'll try to do today is to marry those names with the platforms that they're using to clarify that. I also want to talk about two common things, because I have limited time, that these vaccines have, these vaccine trials have in common. And the first and very important one is the size of the phase 3 trials. So all of the vaccines who are in phase 3 trials now have between 30,000 and 60,000 participants. Now, normally for a vaccine to be approved in the United States, it usually requires around 15,000 participants. So we're looking at 2 to 4 times the normal number of vaccine participants in these trials. And the reason is haste. They're trying to get the number of participant months up quickly, and the N of how many people actually get or are protected from the disease up to a significant amount quickly. But the advantage of having these huge trials is we will have more safety data from these vaccine trials than we will have for almost any other vaccine that we've had. In this talk, I'm not going to dwell on the cellular mechanisms of action for all of these vaccines. And the reason is they have a common final pathway, and I think it could take up a lot of time in this talk. But their final common pathway is the development of or presentation of a spike protein to our humoral antibody immune system and our cellular immune system. And so whether it's via RNA or DNA or preformed protein that's on the outside of a viral-like particle, they all present in the same way. But finally, in this talk, I'm going to talk about safety. And this, I think, is the most important thing for you as providers. I want you to take away from this talk and to reassure yourself that no matter how fast these vaccines are being produced, that the safety steps are not being skipped. And I'm going to go into detail about this. And I want you to know that, so when your patients, your family, or friends talk to you, you can talk in a meaningful way. In that regard, I want to define one thing, and then I'll move on to these 3 questions. And I want to define EUA because it comes up all the time. And at the very end, I'll contrast emergency use authorization versus licensing. But after 9/11, around 2004, when there was an anthrax scare, the FDA was given by law a new power. And that was to approve a medical product that may have clinical benefit in the context of a public health emergency. And already we've seen the FDA utilize this EUA approval process. So I want you to understand where it came from and what it means, and then we'll talk about it at the end. All right, so the 3 most common questions I'm asked when asked about vaccines are, what kind of vaccine will we get and when? How will we know that it works? And will it be safe? So moving on to the next. What kind of vaccine will we get and when will we get it? All of you probably know about Operation Warp Speed, so I will go through it relatively quickly. The US has invested $10 billion in basic science, vaccine development, clinical trials, and manufacturing infrastructure. And they decided to pick 4 vaccine platforms that you see here, and they chose 2 different vaccine makers for each of these sections. So in the mRNA world, the messenger RNA world, it's the Moderna and the Pfizer vaccine. In the non-replicating viral vector vaccine, it's the AstraZeneca or the Janssen. The AstraZeneca is also called the Oxford vaccine, and the Janssen or Johnson & Johnson vaccine. In the protein subunit with adjuvant vaccine is the Novavax, and that will be starting phase 3 trials at the end of this month, and Sanofi Pasteur, which will be starting the following month. For those of you who don't know, an adjuvant is something that's added to a vaccine to augment an immune response. And finally, it's not, Tom pointed out that it's not live attenuated replicating viral vaccine, it's an attenuated. Viruses are not living in the way that we think about living and reproducing, but an attenuated replicating viral vaccine. And Merck is the company that is going to be involved with either one or more of these, and there may be another company that I'm unaware of. But I'm going to focus the ones on currently in phase, the 5 that are currently in phase 3 phase 3 trials. Let's go to the next slide. I'm going to spend a little bit of time on this slide, and because I'm asking the same questions for each of these. I'm going to go through each of these platforms and ask the questions. Do we have precedent for this platform? I'm not going to do how it works because I realized it was going to take too long. Where in the phase 3 trials are we, and when is the expected readout? When will we get to see the data that shows its effectiveness and safety? What are its known advantages and disadvantages, and issues about scalability and manufacturing? This last section is huge. So developing the vaccine is a big process. Going through the clinical trials is a big process, but actually scaling up to vaccinate everybody is another huge issue, and I want you to be aware of what the unique challenges might be for each of these. So for mRNA vaccines, we have no precedent for this type of vaccine, although many researchers around the world are looking at flu vaccines with an mRNA approach or RSV vaccines. And it's because it's easy to adapt. The data, they have the mRNA vaccines, Moderna and Pfizer have enrolled everybody in their trials, and they are aiming for reading out this data this month or next. And my suspicion is, in reading the tea leaves, is that Pfizer may be ready to announce its results as early as the Advisory Committee on Immunization Practice meeting on the 28th through 30th of this month. But certainly Moderna and Pfizer are very close to reporting out the data. And in anticipation of that, Pfizer has been putting out additional information. Some was in the New York Times yesterday. About 50% of their participants are between 50 and 80 years of age. They're starting this vaccine trials on adolescents. So it suggests that they think that they have a successful vaccine platform, and we will likely be hearing about them. And my suspicion is that one of these 2 vaccines will be the first vaccine that we're able to give Oregonians. The advantages of an mRNA vaccine is the RNA doesn't interact with our genome at all. It doesn't mess with our DNA, and that's important. Secondly, it's done very well in clinical challenge trials in primates. In other words, if we give the vaccine to a primate and then 2 weeks later challenge them with a SARS-CoV-2 virus, they don't get sick. And finally, that the mRNA vaccines access its own adjuvant, so we don't need to add something to augment the immune response. The disadvantages, and this is substantial because these will probably be our first vaccines, is that it requires an ultra cold chain. And you've heard about this. but minus 70 degrees centigrade is not something that most freezers can get to. And it makes it transporting and safely delivering all this vaccine quite difficult. We're also not sure, because it's a novel platform, how long lasting the immunity will be. From a manufacturing scalability standpoint, it's probably in the best position. It's a complex, but it's still a semi-chemical production that allows for, that's much, much easier than biological processes. And so it's very scalable. And currently, each of these companies, Moderna and Pfizer, have in the single-digit million dose, so somewhere in the single-digit million dose stockpiled doses, and expect to make 10 million doses of each of these vaccines in both November and December. So that's really important for us to know as we're thinking about anticipating our statewide plan. As far as the non-replicating viral vector vaccines, there have been precedents. Several animal vaccines have used this technology, and the readout is unknown, but AstraZeneca, the two companies are AstraZeneca and Johnson & Johnson. The AstraZeneca will likely be in November or December, and Johnson & Johnson will be January or February. Their advantages are that they don't require an ultra cold temperature, that it's just a frozen vaccine that can be more easily transported, and so we'll have wider providers of vaccine. And the other advantage is the Johnson & Johnson vaccine is the only one of these that I'm going to discuss that is only one shot and not two shots. So not having to follow someone up for a second vaccine is a huge advantage. The disadvantages for both of these is the data safety board, monitoring board. Thank you. The FDA's data safety monitoring board put both of these vaccines on hold. Now, it's not unusual in vaccine trials of this size, 30 to 60,000 people, that people will get sick. And so the job of the Data Safety Monitoring Board is, when someone gets sick, to unblind the study and look at whether or not they got the vaccine. And then if so, then do an investigation where there are other factors that led to this illness. And unfortunately for both of these platforms, they're both on hold. Now, it's not unusual, again, that in a vaccine trial like this, there may be pauses 3 or 4 times in this process. So it doesn't mean that this won't ultimately be found to be a safe vaccine. And it does mean that the Data Safety Monitoring Board is doing its job. From a manufacturing standpoint, it's really interesting. They are both being manufactured in one plant in the United States. And they have not stockpiled large numbers of doses. And as a result, even if they had good data that they could present, the FDA may not accept an EUA authorization request until they have an adequate supply of the vaccine. And so even if they have good data, they may have to wait till they've stockpiled a large number of these vaccines. Regarding the protein subunit with adjuvant, the one you'll hear about is Novavax. It's starting its phase 3 trial now. There are tons of successful vaccines that have made this way: hepatitis B, Haemophilus influenza B, and most recently, the shingles vaccine, the Shingrix. Their data readout is projected to be in March or April timeframe, and its advantages is that it requires normal temperatures, and it may work better in the elderly. These adjuvanted vaccines tend to get a better long-lasting effect for the elderly. The disadvantage is they sometimes cause more local symptoms. With regard to manufacturing, these are probably the most complicated to produce, but they have a huge lead time, and they're expected to have millions of doses stockpiled by the time the readout occurs. Finally, the attenuated replicating viral vector, Merck has successfully done an Ebola virus vaccine that used an attenuated vesicular stomatitis virus to carry the gene for the Ebola surface protein, and this is the same strategy that Merck plans to use. The advantages being that it might be one shot and it could be lifelong, because these are replicated viral vectors. These we won't hear about till the middle or end of 2021.
And so to be clear, Joe, that would be, they would not be live coronavirus. They would be other
That's right.
Weak viruses, I shouldn't say live. Weak viruses that have a little bit of the corona spike protein inserted, so they produce that and then stimulate the immune system, right?
That's exactly right. So they use it as a carrier virus that's replicating in their DNA. They've genetically engineered that DNA to code for the spike protein. So when I look at how things, whether it's going to work, I look at the preclinical studies and especially the primate challenge studies, because they really reflect whether or not it's likely to work in humans. In the phase 1 and 2 trials, these are human trials with small numbers. The things to look for are going to be what are the levels of neutralizing antibodies. And that's compared to people who have had these infections naturally. And there should be orders of magnitude higher. So they'll look at the humoral, the antibody response, but they'll also look at the cellular response. And in general, they check both T helper type 1 and T helper type 2 cells. And they've found, and we can talk about it if the people are interested in the question and answer, why it's important that they have a T helper type 1 predominant cellular response. So those are the things we'll be looking for in those early phases. And finally, the phase 3 trials are obviously the most significant and most important. And they have to get to an N of about 150 cases. That would be the ideal, to know whether or not this vaccine is going to work or not. They generally are doing it in a 1-to-1, meaning 1 placebo person to an active vaccine, or 1-to-2, 1 placebo to 2 people receiving the vaccine. And an excellent endpoint would be 150 cases. They should be able to tell whether or not these are effective. Now, almost all of the vaccines are aiming for 60% effectiveness, except for AstraZeneca, which is aiming for a 50% effectiveness. Okay, next slide. So this may be the most important slide for you to take away today. Will it be safe? I want to emphasize that the vaccines are the most regulated medical products in the United States, more than any medicine, more than any procedure, and that there are a number of safety steps that there are remaining in place, even though we're moving quickly. And one thing that helps us with it that I've mentioned before is that the vaccine sizes, the trial sizes are 2 to 4 times larger for COVID vaccines compared to normal vaccines. And so we will get a lot more safety information. Last week, the FDA changed its rules to make it more strenuous to consider an emergency use authorization. They said that the manufacturer had to wait 2 months from when the vaccines were completed. So we'll get 2 months of just watching to see if people get late effects. Now, it's not 3 months or 4 months. There are probably going to be some adverse effects that happen very far out, but they're very, very rare. I also want to point that there's 3 levels of independent oversight by non-governmental, non-pharmaceutical industry sciences that are going to look at this data before the FDA gives its approval for an EUA. The Data Safety Monitoring Board, which I mentioned earlier, their job is they're the only ones who can unblind these studies. They can look for effectiveness, but they can also look if there are adverse events. They can look to see early whether they need to pause or to stop a trial. Then there's there's a Vaccine and Biological Products Advisory Committee, and this time they're going to have an open session for public scrutiny so that everyone can see the data that they're going to be using to make a decision and recommendations to the FDA. And finally, the Advisory Committee on Immunization Practices will give the final say about how they recommend the FDA uses these vaccines. All of these safety steps are still in place. And I think we need to be reassured that this is far and away more than other countries are doing. A lot of countries are doing 2,000 and 5,000 people studies. We will get a robust amount of information. And I think if a vaccine comes on the market, then nothing's 100% safe, but it's as safe as other vaccines that we've been given and we continue to take regularly. And the last thing is is what's the difference between an EUA and a full FDA license? There's good and bad, right? So an EUA saves time. I've been told that it's tens of thousands of pages that have to be put together for a licensure, and it takes months. This is a pandemic. The time is critical. If we're going to move through this, we need not only hand washing, social distancing, and masking, but we need a vaccine. And so time is of the essence. And while with an EUA, we won't have ultra long-term, more than 2 months safety data, we will at least have that 2-month safety data to look for the vast majority of adverse effects associated with these vaccines. So some of the downsides, when we release a vaccine through an EUA, we won't actually know if it actually prevents transmission as well as preventing disease. In other words, if I get the vaccine, I may not get sick, although I may still be infected and I might be able to transmit. And we will find that out over time, but we won't know for sure when this gets approved. Early release of any vaccine might adversely affect other ongoing trials from an ethical standpoint. So if Moderna or Pfizer vaccines are found to be safe and effective, and a patient of mine is in another trial for another vaccine, is it ethical to keep them blinded to the fact that they might be getting the control? And so other manufacturers may have to look at whether that gets unblinded to offer those people the vaccine that's known to work. And this is going to slow down other, maybe better vaccines underway. And if a vaccine is accepted under EUA, then in the future vaccine trials, that will have to be the control, the vaccine that has been approved under EUA, and that the new vaccine will have to prove equivalence or improvement over that previously approved one. So I think that's all I have to say for today, and we may have questions.
Yes, thank you so much, Joe. So first of all, there's just a lot of comments about how useful this has been, so thank you. And also a request for making this a regular part of our future webinar topics, maybe in not as great detail every time, but certainly updates. So thank you. Also noting a couple people have asked to include the names of the brand vaccines as you shared verbally under each of the types of vaccines, just for them when they get the slides. So we can do that before we post them, so that people have that reference. And just a reminder, all of these slides, as well as the recording of these webinars, are posted on a healthcare provider website, our COVID provider website, and I shared the link to that on the broadcast. So hopefully you've all been able to locate that previously. So a couple questions that have come through. Maybe you can speak a little bit to the prioritization during early rollout. We can also highlight that at a future webinar. There was a question about what guidelines are being discussed by OHA, our agency, regarding providing prioritization of vaccines. The National Academy of Sciences has just published comprehensive recommendations. ACIP will be taking this up. Can you share just a little bit about how OHA will approach those?
Yeah, I think we're looking at both of those documents, and ACIP hasn't put out its document, but they will on that 28th through 30th meeting. And I think the 1A group there's relatively uniform agreement on what that might look like. And those are people who are at high risk because of oftentimes the work that they do. So we would be including healthcare workers, whether paid or unpaid, and their support staff that support them because they are at equal risk, be it in hospitals or clinics as well. When it gets to 1B, it's going to partly depend on the quality and characteristics of the vaccine. So 1B group may be people at higher risk. So it may be the elderly, or it may be people in racial or ethnic minorities. But the data from the vaccine trial will have to reflect that it's effective in those groups. So the 1B and how we prioritize may be affected by what the vaccine looks like and what the data around effectiveness in these subgroups are going to be. But in general, the 1B group are essential workers who keep our society working. And it's a broad group of people. But but we will likely follow the recommendations of ACIP largely. That's what we've done in the past, but we will look at our own unique Oregonian situation and make tweaks. We will have that opportunity to make tweaks within that, but I think that's what we'll do.
Great, thank you. And we will, as more information rolls out, particularly on the prioritization, timeline, and phases, we will be sure that's part of our future webinars, but great question. To that end, I'm going to just insert maybe a question that you can share, and I can speak to just briefly. You know, we know that vaccine hesitancy has been part of Oregon for a long time. And of course, there's different uptakes even of influenza vaccine among different populations. And certainly there's anticipation of hesitation both across healthcare providers and as well as different parts of our community members with regard to a vaccine. And I can just say and see if you have anything to add, Joe, that we are doing a lot of work through our communications program with communications teams representing different cultures and language to try to get effective information out there that meets the needs of Oregonians as they decide on the COVID vaccine ahead. So we know this is a really large part of this work. And so more to come as that work is underway. But anything you'd add to that?
I couldn't agree more. I think this is a really critical thing. The CDC just came out with a study looking at healthcare workers and how they felt about the vaccine. And about 50% of them said that they were hesitant. Not that they wouldn't take it, but that they were hesitant. And the things that they said, the messaging that they said really made them think more about getting the vaccine included: stopping the pandemic is going to take all of our tools, hand washing, masks, social distancing, and vaccines. Taken together, these tools offer the best chance of getting our communities and schools and health systems back to normal. Number two, that COVID-19 has a huge impact on daily lives for many people. A COVID vaccine can help you stay healthy, give you peace of mind, and get you back to spending time with family and friends. And number three, your work is important. You take precautions to keep yourself and your family safe, especially now during the pandemic. Don't bring your work home with you. Get the COVID vaccine. And the following safety messages, again, emphasize what I said, that no steps are being skipped in the clinical trial data, and that no matter how quickly the COVID vaccines are developed, they're being held to the same standards as other vaccines.
Thanks. And just one more comment based on someone shared about the level of concern, particularly across BIPOC communities. And one additional thing that we'll bring forward as this work continues to unfold is that as we pull together an advisory committee and other work streams related to COVID-19 vaccine planning, we are really focused on health equity, really centered in this work, both in the planning and development and implementation, and really plan to have diverse representation in the work that we're doing on planning to make sure all of those perspectives and input on this developing work are really centered for the COVID vaccine. So more to come on that. I know that there are a lot of questions here. We will make sure that we incorporate them. There's some additional questions around safety. There's some questions about the particular way that some of these vaccines work. So we will capture them all and incorporate them into our future webinars and information that we put out. I want to make sure we have just a few minutes on the next topics before we run out of time.
Can I ask a quick question?
Yeah.
Do you think, or what's OHA's thoughts on the timing of getting a vaccine out, just looking at how trials are going?
I think that that we could see vaccine definitely in December. I don't have a crystal ball, but from what I'm hearing, and Mansaf Salawi is the head of the Operation Warp Speed. He just gave a thing at Johns Hopkins 2 days ago saying that it's likely that we will have vaccine. And so I'm assuming this is an mRNA vaccine, but I don't know exactly, but it's coming pretty quickly.
Great. So just a couple slides, knowing that we are now entering flu season, and this is particular importance, always is important and particularly relevant during this COVID-19 pandemic. So this is just a couple slides we pulled from our flu bites. This is some of the epi surveillance data we do on flu, and you can sign up to subscribe to OHA's Flu Bites and have that come in, I believe, weekly in your email. So we've included that below on the slides as a link or information on where you can access this data. But just to share, we are early on in flu season. You can see this is data at a glance. So last week, we've been able to report data from September 27th to October 3rd. This data shows in the top line percentage of emergency department visits that we've been monitoring also for COVID. So this is influenza-like illness. You can see here that there's maybe the slightest uptick from the last week. The percentage of positive influenza tests, this is the area that we'll be monitoring closely throughout the season. Right now it's at 0.3%, and I'll show you more information about that. As you can see, we've added information about COVID-19 hospitalizations to our flu bites as well. Next slide. Again, this data is still so early because we are early in the flu season here. But this looks at the different regions and the tests that have been done in the second column for flu. And then the number and percent that are positive for both flu A and B. So you can see here we've only had one positive flu test in Oregon reported up through October 3rd, and that was for flu B in the Southern Oregon region. And then this is actually even newer data. I was able to get a hold of this just this morning. This will be, I believe, part of our next flu bite. The current data on our last flu bite, that's the one posted right now, does not have this most up-to-date information. And this is just showing the flu vaccine doses given as submitted through our alert system by week. So the bright blue line is for this current flu season, and it's comparing to 2019 and 2020 season in green, and then the yellow is 2018 to 19. And we're glad to see that it appears that we're having an increased uptake in flu vaccines compared to past flu seasons. And we'll continue to monitor this actively. In addition, we can look at flu immunizations by age in Oregon. Again, this data was from up through the week of September 27th through October 3rd, so a little bit older than the one I just showed you. And fortunately, we're seeing a higher uptake of the flu immunization compared to last flu season across the majority of age groups, although the area that we're seeing the most increased uptake compared to 2019 is in the 60 and above population, or 55 to 80 or so year olds. So we'll keep monitoring. We are seeing increases even in children, a little less so in the young adult age, but we will continue to monitor this, and overall are feeling good that we're seeing such higher uptakes of flu vaccine this year. And then briefly, we're going to have Dr. Smits go through some of these literature updates for you. Go ahead, Dr. Smits.
So I'm just going to give you the highlights of a couple studies that I pulled out. This was a point of care and rapid testing review, Cochrane review that was just published. So these are, this kind of goes along with the BinaxNOW and those other tests that Dr. Jeanne was talking about. They looked at 8 study cohorts and found that the antigen, so this would be just looking for pieces of virus rather than the viral RNA, sorry. The sensitivity varied a huge amount, with the average sensitivity being about 56% and the average specificity about 99%. So if, just as Dr. Jeanne was talking about, if you get a negative test, it's almost a coin flip. You need to still continue to quarantine. The rapid molecular assays are more sensitive, about 95% with almost 99% specificity. So a little bit better.
And these are all compared to PCR?
They're all compared to PCR. Yeah. So 56% of 80% is the antigen test. So yes, like I said, about a coin flip. Next slide. And so the conclusions and the Cochrane from the Cochrane team was that they were uncertain about how these would actually work in clinical practice, and they were very concerned about their use in asymptomatic people, because if you have a 50% sensitivity and you are asymptomatic, we know that the sensitivity is even lower in that group. So a lot of concerns there. And they thought maybe these would be good tests to be sort of a triage. So if you have a positive on these tests, it's probably a positive. If it's a negative, maybe then you could go on and use the PCR, which takes longer.
And just to clarify, that is not currently our active recommendation, but we are looking actively at all the data coming in, particularly on these new test platforms, and doing also some additional testing ourselves to figure out how that informs our ongoing guidance. Is that correct?
That's right. Yeah.
This was the Cochrane review guidance, not OHA.
Yeah.
Thank you for that clarification. I just wanted to highlight this new systematic review that came out in BMJ this week. It's a large study, had about 15,000 patients, and it just showed that how high the rate of acute kidney injury was in the critically ill population. And so if you're doing hospital medicine, I just wanted to pull this study out so that you are aware that not only do really ill COVID patients have a high rate of kidney injury, but they actually had a much higher rate of mortality when they had that kidney injury. So to make sure that you are definitely monitoring and providing adequate support for their kidneys when they're in the hospital. And then the last study I pulled out was, there's a lot of things going on in the press right now about whether COVID is actually causing any increase in deaths, or whether we're just seeing people who would have died of other causes dying of COVID. I get some of this with my own patients. So this was a really good study that just came out in JAMA, and they found a 20% increase in expected deaths between March 1st and August 1st compared to last year. So 20% more people died than you would have thought should have died. Of those deaths, 67% were due to COVID-19. And then the other two groups, the last third was divided into other two groups. One was increase in heart disease deaths, whether that's because people weren't getting adequate 911 responses or they weren't going to the hospital because of COVID fears, who knows? I mean, we don't know for certain, but definitely, or maybe COVID has some type of heart damage that's been proposed as well. And then increased mortality rate for people with Alzheimer's and dementia, probably related to the fact that these folks are more likely to be in nursing homes, and we know that that's a high-risk area. That was it. So that's the answer to that question, to the question of whether this is actually increasing our death rate. It is.
Mm-hmm. And Tom, you mentioned a data for Oregon that was even higher than that. Is that right? You said 28%? No.
Well, lower than that study, which in terms of the percentage that was COVID-19 related.
Oh, okay.
Yeah.
And what was that? I'm sorry.
Well, it's on our main healthcare provider website. We have dashboards on death data, both by weekly expected deaths and the percentage increase. Oregon, by and large, has seen fewer weeks where we've had significantly increased death over expected compared to the 5-year average. And then to date, we've had about, I think, 29% of the excess deaths would be explained by COVID deaths, but that's definitely an underestimate, because it just comes from death certificates, and some people who died from COVID may not have been diagnosed. So
I understand. Okay, thank you.
We can pull some of that Oregon data into one of our future webinars to clarify that more, but thank you both. Quick topic, thermometers.
So this came from a clinical question from our last seminar. And it was basically a question about using forehead thermometers. How effective are they? And the context was, in a clinic, you have a higher exposure if you're using an oral thermometer. So is it worth the higher exposure risk versus a forehead thermometer? So there's been a couple of good systematic reviews done. This is obviously a hot question right now with COVID. The Oxford Center for Evidence-Based Medicine, I would actually advise anyone to go look at that website. It's great. They have tons of different different rapid reviews, but they did one and found that the chemical forehead, so those would be like those chemical strips, basically are horrible. They don't detect anything. You just can't use them. The Canadian technology group actually did a review about 6 years ago looking at 20 studies, and they compared the handheld infrared skin thermometers to rectal or nasopharyngeal, like all the other things that you normally do, rectal, oral, axillary. And they found that the sensitivity was a lot lower, 76 to 97%, with the specificity was also lower, 79 to 99. And their positive and negative predictive values were lower with the infrared skin thermometers. So the next slide. So basically, looking at at this and some other studies that I didn't have a chance to put on here, forehead thermometers are really not that accurate for diagnosing fever. And if you are really trying to measure somebody's temperature, you need to do oral, rectal, axillary, the more classic. So if in your clinic that is something that you need for your clinical decision making, it's worth a slight increase in exposure.
One question that came up, just to clarify what peripheral thermometer is. Sorry, we'll go back. Verdict, peripheral thermometers are used, they don't detect fever.
Oh, so peripheral thermometers are anything that are not like put into a orifice. So there's tympanic membrane thermometers. They also don't work very well. Forehead thermal scans, and then there's these scanners that you can, I don't know, walk through or something. There's different technologies. So it's basically trying to not really come in contact with somebody, but still get a temperature reading.
And how about, does that differentiate from axillary, or do
Technically, they're counting axillary as in the same category as like oral and rectal. So yeah, you're right. They overlap. The axilla is peripheral, and you could say the tympanic membrane is more close in, but still.
Okay, well, we are at time. We got some really good questions over the course of the last 20 minutes. We will look at responding to all of them and your requests for more detail in our slides, particularly about vaccines. So stay tuned. We'll keep you updated, and we know there will be more and more information coming. So thank you to the team here for all this work, and for everybody listening for all the work that you are doing on the front line as well. Take care and have a good day. We will be joining the Project Echo webinar next Thursday, and we'll be back with you in the weeks following. So have a good day. Thanks, everyone.